All right, BIM 100 crew, let's get into it. This is your crash course for Professor Pasquinelli's final. We're going to focus on what really matters, cracking the code on all that experimental data. You know, the stuff straight out of Lecture 13. So, you ready to do this? We're going to break down exactly how to tackle those data interpretation questions. You know, the ones I'm talking about. The goal here is to give you a solid game plan so you can walk into that final feeling confident. Okay, this is super important. Pasquinelli's final isn't about just spitting back definitions. No, the whole point is to think like a scientist. It's not enough to know the name of a technique. You've got to understand what the data is screaming at you about what's actually going on inside that cell. All right, first up, the chromatin story. Think of it this way. Before a gene can be read, the cell has to be able to get to it. Is the book open on the right page, or is it slammed shut and locked away? That's what we're trying to figure out. So, how do we check if the book is open? The go-to technique here is the DNase eye protection assay. It sounds complicated, but it's actually pretty clever. We use this little enzyme DNase that basically acts like a pair of scissors just chopping up DNA. The whole idea is to see if it can get to our gene of interest. And here's the bottom line, the pro tip you need to remember. If the chromatin is open, we call that euchromatin, then hey, the gene can be transcribed. But if it's all packed up tight, that's heterochromatin, it's pretty much silent. It's off limits. Simple as that. Okay, let's get our hands dirty and look at a real example, probably something just like you'd see on the exam. Don't let the bands intimidate you. There is a super straightforward step-by-step way to read this thing, and once you get it, you can solve any of them. Step 1, and this is non-negotiable. Always find your controls first. Before you look at anything else, find the lane where they didn't add any DNase. You see that nice thick band? That's your baseline. That's what the DNA looks like when nothing has touched it. It's your 100% mark. Okay, now for step 2. Look at the other lanes, the ones where they did add the DNase. The whole trick to reading this gel is just comparing the intensity of the bands in these lanes to that control band we just found. That's it. So, as you're looking at each lane, you just have to ask yourself one simple question. What happened to the band? Is it still there and strong? Or did it get fainter or maybe disappear altogether? And this is how you put it all together. If that band is faint or totally gone, what does that tell you? It means the DNA scissors got in and did their job. The chromatin had to be open. On the flip side, if the band is still looking strong, just like your control, the DNA was protected. It was hiding inside that condensed chromatin. See? Not so bad. Awesome. So now we know how to tell if a gene is open for business. But that's just part of the story. The next question is, who's showing up to work? Which proteins are actually binding to the DNA? Let's check it out. Right, so when we talk about proteins binding to DNA, there are two big players you absolutely have to know for the final. EMSA and CHIP. They both look at protein DNA interactions, but they do it in slightly different ways. Here's the key difference. Think of MSA, or a gel shift, as a very specific test in a tube, in vitro. It asks a super focused question. Does this protein stick to this little piece of DNA? CHIP, on the other hand, is an in vivo investigation. It's like asking a much broader question. Inside a real living cell, where in the entire genome is my protein hanging out? Let's talk EMSA first. You're running DNA through a gel, which is kind of like a race. A small piece of DNA can zip right through. But what happens if a big, clunky protein grabs onto it? Well, the whole complex gets bigger and heavier, and it's going to move way, way slower through that gel. And that leads us to the Pasquinelli pro tip for EMSA. A shift means something is bound. When you look at the gel, you're looking for a band that's higher up than the band for the DNA alone. That higher band, that shift, is your smoking gun. It proves your protein is stuck to the DNA. Okay, now let's switch gears to CHIP. This one's pretty cool. Imagine you have a special magnetic fishhook. That's your antibody. And it only catches one specific type of fish. That's your protein of interest. You dip it into the pond of the cell nucleus, and you pull out your protein. And guess what? You also pull out whatever it was holding on to, which in this case is the DNA. So after you do this pull down, you analyze the DNA you caught. If you see a really strong band on a gel or a big peak in sequencing data, that's your answer. It means there was a ton of your protein bound to that specific spot on the DNA inside the living cell. All right, we've covered whether a gene is accessible and who's binding to it. Now for the grand finale. What's the actual result? Which genes are ultimately turned on or off? Let's zoom out and look at the big picture of gene expression. The tool for this is the microarray. Honestly, the best way to think about a microarray is as a massive census of all the genes in a cell at one specific moment. It lets you ask for all 20,000 genes, are they more active in condition A or condition B? It's incredibly powerful. So check this out. This is what one looks like. In this experiment, they took cells grown with serum and labeled all their gene messages red. Then they did the same for cells grown without serum, but labeled them green. They mixed them all together and poured it over this chip. The color you see in each little dot is the punchline. And reading the colors is actually super intuitive. If you see a red spot, that gene is cranked up in condition 1. If you see a green spot, it's cranked up in condition 2. What if it's yellow? Well, red and green make yellow, right? That just means the gene's expression is pretty much the same in both. And a black spot? That gene's not being expressed at all. Simple.
Okay, so those are the key techniques. Now, for the most important part, how do you take all this and turn it into a surefire strategy when you're looking at a big, scary data question on the exam? Let's create your game plan.
Here it is, your 4-step plan for success. It works for any of these problems. Number 1, first, just identify the technique. Ask yourself, what am I looking at? A DNA assay, an EM assay? Number 2, find those controls. I can't say it enough. Number 3, look for the result. Is there a shift, a color change, a missing band? And finally, number 4, tell the story. What does that result mean biologically? Just walk through those steps and you'll nail it. Look, every gel, every microarray, every piece of data tells a story, and you now have the tools to read it. Don't just see bands and spots, see the biology happening behind them. Go into that final with confidence. You got this. Good luck!